So I'll go ahead and get us started. First of all, thank you everybody for being here. I'm Joel Briere, and this is Victoria Wushner, and we are with 5-MeO-DMT Information and Vital Education. And we are happy to have Manesh Girn with us today. And just pull up bio. So Manesh is in the latter stages of obtaining his PhD in neuroscience at McGill University and has been a lead or co-author in over a dozen scientific publications and book chapters on psychedelics, the default mode network, and related topics. He actively collaborates with leaders in the psychedelic space, psychedelic science, such as Dr. Robin Carhart-Harris, and is also Chief Research Officer at a Canadian psychedelic bioscience company, EntheoTech Bioscience. Finally, Manesh runs a YouTube channel and Instagram page called The Psychedelic Scientist, where he discusses the latest topics in psychedelic science in an easy to understand but non-superficial form. Manesh, thank you for being here today with us.
Thank you so much for coming.
Yeah, really truly my pleasure. Yeah, thank you for the introduction.
Absolutely, absolutely. So, today we're going to be talking about boosting neuroplasticity with 5-MeO-DMT. And Manesh, we're really excited to hear your take on this. The default mode network is a very tossed around term in the psychedelic space these days. And it's always nice to get some real good knowledge and some real good understanding around what it really means and how it correlates with the psychedelic experience.
For sure. Yeah. Can I take it away or is there more intro?
Absolutely. Take it away.
Okay, great. So today what I thought I would do is I have a little presentation to get us started. And then after that, just open it up like a dialogue with you two, take questions from the audience or whatever makes sense. I think that's often where a lot of the great discussions happen in discussion because then it's more relevant and like cater towards what people are looking for. But so I think I need to be enabled to share my screen.
Gotcha.
Share screen.
No, that's for
that should work. Give that a shot.
Yep. Okay. Okay, you can see this, right? Cool. So so the topic of this is 5-MeO and neuroplasticity, but I thought in order to really describe it, I should give some background and show the broader kind of context in which these findings exist or this research exists. And so just to make sure we're all on the same page, I'm going to first drop in with some basic neuroscience to understand even what neuroplasticity is, to provide a foundation for that. Then dive into what is the definition of neuroplasticity, because it seems like a lot of people have different conceptions of what it means. And it's actually an umbrella term that can refer to a lot of different things. And so I'll talk about that. Then I'll talk about this really strong link between neuroplasticity, chronic stress, and mental illness, whether it's depression, PTSD, or addiction. These three things go hand in hand in a variety of different ways, which I think are interesting and relevant for understanding how psychedelics can help with these kinds of things. And then I'll talk a bit about how 5-MeO is actually quite different than the other serotonergic psychedelics. And by serotonergic psychedelics, I'm mainly referring to psilocybin, LSD, and then DMT. As well as ayahuasca. And then I'll talk about the neuroplasticity research specifically on 5-MeO-DMT, and then talk about it in relation to the serotonergic psychedelics, because there's more research on them in terms of neuroplasticity, and there's reason to think there's a lot of similar things happening with 5-MeO that hasn't necessarily been found or studied with 5-MeO yet. And then I'll end with some takeaways, and then we can chat and have a little Q&A. So some basics. So the brain is composed of around 100 billion brain cells called neurons. And the interesting thing about neurons is they don't physically touch. Like a subset of them do, but let's not go there. There's a small percentage that do physically touch, but the majority don't. So then it's like, okay, so presumably these brain cells, these neurons are communicating. How do they communicate if they're not physically touching? And so they communicate through chemical signals called neurotransmitters. So I'm going to walk us through this little diagram here. So in the small here, these are two neurons. And you can see there's this blue one and then a purple one. And there's a long kind of protrusion from the cell and then little branches at the end, which then attach to the branches of the other one. And so where these things intersect is this thing that's enlarged here, and it's called a synapse. And so the neuron on the left which we call the presynaptic neuron, like pre-synapse, before the synapse, is releasing neurotransmitters, these little bubbles of chemicals, into the middle, into the synapse. And then that kind of moves across this empty space, this very small empty space, and can then latch on to receptors in the postsynaptic neuron. So the purple one here. And these things here are receptors, and the neurotransmitter comes out, it gets stuck to the receptor, and then can open it to allow different molecules to come into the cell to change its charge, to make it fire, basically. So how neurons generally affect each other are through the presynaptic neuron releasing neurotransmitters into the synapse and then affecting receptors on the receiving side, postsynaptic. And so keeping that in mind, well, I guess, yeah, here's an analogy, is that each given neurotransmitter only is able to attach into particular receptors. And so each neurotransmitter can be seen as a key and each receptor is a lock, and not all keys are going to fit into all locks and so on. And that's kind of how it works in the brain. Now, when we talk about neuroplasticity, we're talking about the strength of these synapses, strength of connections between two neurons. And the strength of a synapse can be altered in a variety of different ways. So this could, for example, be on the presynaptic side, that could release more neurotransmitter, which then affects more receptors on the right. Or the right could just have more receptors in general. So there's more places where these neurotransmitters can land. And those are just two, but there's many different ways the strength, so the ability of the presynaptic neuron to affect the postsynaptic neuron can be altered and changed and strengthened or weakened. And in addition to that, a given postsynaptic neuron's ability or tendency to fire is based on all the different presynaptic neurons that innervate it or that connect to it. And so it's like all the synapses combined on a given neuron determine whether it's going to fire or not. And on the purple neuron here, where it's receiving this blue neuron, these branches are called dendrites. And this is an important point. So dendrites are attached to the cell body of a neuron and are the receiving station of a neuron. So this is where it receives inputs from other neurons. And as I said, usually a given neuron actually can receive up to 10,000 inputs from 10,000 different other neurons. And then somehow it takes all 10,000 of those and just determines whether it fires or not. So it's extremely complex. But if a neuron has more dendrites, more of these receiving stations, it can receive more inputs. So more other neurons can influence it. And so the complexity of that neuron's firing goes up if it has more dendrites, because now it has to take into account the firing of all these different neurons, which again can be up to, which on average is 10,000 and can be higher than 10,000. So that's some basics of neuroscience. I went by pretty quick. Hopefully a lot of you had a bit of this knowledge, and I hope it's clear. And okay, now let's use that as a jumping off point to talk about neuroplasticity. So we can give a very general definition for it. It's kind of processes that support the creation of new connections between neurons, so new synapses, and or the alterations of the existing ones. So either creating new synapses on, for example, the dendrites of a receiving neuron, or altering the ones that exist to make them strong or weak, or removing some. And this process happens all the time. And it's essential for learning and memory. How else would our brain encode new things we're learning if the neurons are not changing? So they change in their connections with each other to support our ability to learn new things and just our ability to remember things in general. And as we adapt to new environments and experiences in our life, our brain needs to be able to change to support that. You know, if you have an entirely new, let's say you enter an entirely new environment that you've never been in before, you don't want to just bring everything you were doing from other environments and apply it there. You want to be able to adapt to it. And that underlying that is your brain's ability to change its connections to enable new ways of thinking and behaving. And then also, if you're injured, if you have a concussion or a traumatic brain injury, the brain needs to be able to kind of reconfigure itself to compensate or recover from that injury. So if a part of your brain is damaged, other parts of your brain will take on more of a load to try to cover for that region. And that's enabled because the brain is plastic in this way. It's able to change its connections between neurons. But it's not always good. Neuroplasticity isn't always good. And I want to say that again, like just because you boosted your neuroplasticity doesn't mean that you're going to be better or healthier, or it's going to be a positive change. For example, neuroplasticity gone wrong happens in chronic pain, because chronic pain happens when there's nerve damage, and then neurons adapt and think that your pain is always there, and so they're firing as if there's a painful stimulus there, when really the tissue has recovered. There's no pain that should be there, but the plastic changes have somehow persisted to keep that pain going, even though when it shouldn't be going, because what it's trying to signal is no longer happening. And then addiction. Cocaine really boosts neuroplasticity in specific parts of your brain. This is what makes it so, like if you do it progressively a lot over time, as well as a whole variety of other drugs, it strengthens those circuits that lead from seeing or thinking about it to, grabbing it and ingesting it in somehow. And those circuits which motivate these habitual kind of impulsive behaviors become very strongly impulsive because of neuroplasticity. And just more broadly, any bad habit you have in terms of your thinking, where your attention goes, your behavior, as well as good habits, but bad habits too, you can just encode them by repeatedly doing them. And given neuroplasticity, you can get stuck in them, and then it's hard to disentangle. It takes a lot of effort to then disentangle and create new circuits, which of course psychedelics can help with, just kind of what we'll talk about a bit later. So that's neuroplasticity. And it can be actually broken down into two different main types. So one is structural plasticity. So this is like actual physical changes. This is kind of what I've been focusing on so far, mostly pertaining to dendrites. So the receiving part of a neuron, the branches that allow inputs from other neurons to hit it basically and create synapses. So the dendrites can change in how many there are, the shape they're in, the size of them, and the number of spines, which are basically landing pads. And so there's very physical changes that can happen to change the amount of inputs and the nature of inputs it's received. And this corresponds to postsynaptic, as I mentioned. And here's a little picture to help with that. I mean, the left, you see I hope you can see my cursor. So at the bottom here is the cell body, the circular thing. And these are the dendrites. So there's a receiving station. And then you go down the axon, as it's called, and these are the parts that hit other neurons. And so this is a one with not many dendrites and not many ways for connecting with other neurons. And then this is one where there's more branches, and each of these can go to a whole bunch more neurons. And so you could see how physically it's changed to enable a greater number of connections. Now, this is kind of getting ahead, but with psychedelics, a lot of these are created temporarily, but which ones stick around depend on what you're doing, right? If you're consistently doing behaviors that support a given connection, it's going to stay. But if you don't, then that's not going to stay, and you're going to go back to this or a slightly altered version of this, which is why integration is so important. These connections aren't magically going to change. You then have to tell your brain that, yes, this new possible connection is an important one. We should keep it. And you do that by repeatedly doing the behavior that activates it. So that's how you maintain it. And slightly different from these structural changes to neurons is something called functional plasticity. And this, rather than corresponding to the physical nature of a neuron, it corresponds to what we might say we could just say the strength of a synapse, the likelihood it's going to fire, the ease with which it fires. And by fire, I mean activate. And all these things related to its ability to conduct electrical impulses, which again, corresponds to the activation of a neuron and the releasing of neurotransmitters. And so a given synapse's strength, so that is a given connection between two neurons, can change due to structural changes, as I show on the left, changes in neurotransmitter release on the presynaptic side, and changes in receptor numbers. All of these can change how two given neurons interact. And yeah, this is based in presynaptic and postsynaptic. So one is structural, like physical, like what is available for two neurons to connect, or many neurons to connect to one neuron. And the next one is functional, meaning it relates to function, the activity of it, and how the strength of its activity and its ability to fire can be changed. So these are two different things. And so why is all this relevant? So what's interesting is that there's a lot of research showing that chronic stress, occurs in any mental health condition. It can either be a cause of one or be caused by it. If you think about it, if you're extremely stressed, your brain responds in a particular way that makes you more susceptible to things like depression. And then if you get depression, you often become more stressed, your stress system's hyperactive, and that could just become a reciprocal feedback loop that keeps you stuck. And we know from work in rodents that if you stress them out for long periods of time in chronic stress models, leads to the atrophy of synapses in the prefrontal cortex and hippocampus. So what do we mean by atrophy? If you look on the left, it's a little example. On the right, this given neuron has less branches, it's a bit smaller, it's less complex, and compared to a healthy one. So really, it kind of shrivels up, loses ability to connect to other neurons, and just loses ability to function effectively. And this happens very reliably in chronic stress. And it happens in the prefrontal cortex, which is a really important, let's see, do I have a figure here? I do. Wait, I'll actually just get to that. So prefrontal cortex, I'll just say, is a very important thing in terms of our ability to regulate our emotions and think clearly. And hippocampus is very important for our memory. So these two areas seem to be particularly vulnerable and correlated with mental health conditions. And yeah, when I say atrophy, this is a bit more detail. It's all these things are reduced. You know, the dendrites basically become, there's less of them. They're less long. Their shape is different. They're smaller. There's less receptors on the neuron as well to receive inputs from other neurons, and the synaptic strength is reduced. So overall, the efficiency of these neurons to receive and propagate inputs is reduced, and that leads to dysfunction in the ability for those brain regions to do what they should be doing. In this case, you know, prefrontal cortex and hippocampus, which are two very important regions. And yeah, plenty of evidence directly correlating these changes to depression in rodents, but a lot of this stuff has been shown to translate as well. And yeah, and here's an example of that translation is like human patients with major depressive disorder, MDD, literally have smaller prefrontal cortices, smaller prefrontal cortex and smaller hippocampus. And as I mentioned, this is particularly, the prefrontal cortex is particularly important for things like our ability to regulate our emotions and kind of guide our thought flow, I think more importantly, and relates to our ability to escape cycles of rumination. And then our hippocampus is involved in our ability to properly encode memories of experiences, and also in kind of connection with other regions, modulate our stress responses to experiences as well. And so these are particularly susceptible to the negative effects of chronic stress, making these brain regions less able to do what they're supposed to be doing, and thereby later on leading to things like depression. And so this whole idea about psychedelics is that they're what we can call psychoplastogens, that they boost plasticity psychologically, but also, you know, neurophysiologically. And so here's another reiteration of what I was just saying. Healthy brain looks like this. Chronic stress, depression, you know, it becomes a bit less complex, a bit shriveled up, a bit less able to engage in its functions. And then findings with psychedelics, which includes psilocybin, DMT, 5-MeO, ayahuasca, ketamine as well. And to some extent, probably MDMA, although where that happens and how is very different, seems to restore those connections that are lost given a mental illness, and literally reverse the negative damage of stress and kind of chronic mental illnesses. And so before I get into some findings with 5-MeO, and I apologize for the baby screaming in the background, nothing I could do about that. I want to contrast 5-MeO with psilocybin, LSD, and NN-DMT, or quote unquote normal DMT, as people might call it. What's interesting about 5-MeO is that the main receptor it hits is the serotonin, or 5-HT1A receptor. Whereas with psilocybin LSD is primarily it acts through the 5-HT2A receptor. So like in this fundamental pharmacological mechanism of action, 5-MeO is quite distinct from the other psychedelics. It even, like, studies have shown it has 300 to 1,000 times more affinity, meaning it's stickier to that receptor than the serotonin 2A receptor, which the other psychedelics primarily hit. But that's not to say it doesn't hit the 2A. What's interesting is the 2A only starts to get hit at higher doses. So you need a higher amount of 5-MeO to start tapping into the kind of the functional and structural effects of serotonin 2A receptors. And yeah, it's the opposite for these other drugs. And 5-MeO, as we know, is very short lasting. And these are predominantly long lasting except and then DMT. And I really want to emphasize this point again. 5-MeO hits the 1A receptor, which is located actually in different parts of the brain. It overlaps with the 2A, but it predominantly located in different parts of the brain, plays a very different function in the brain. And therefore suggests that 5-MeO is doing something fundamentally different, particularly at lower doses than the other psychedelics. But again, it does tap into more of these similar processes at higher doses. And you might be wondering what doses. We don't really know that. We just know that as since it's more sticky at the 1A receptor, it'll go there first essentially, and only later will it start to saturate these other receptors. And so a bit more on this distinction between the 1A and 2A receptors. I think it's informative because it tells us where the neuroplasticity changes are likely happening. So we're going to talk about the 1A versus 2A again. So if we look at this little image here, the 5-HT1A, the serotonin 1A receptor, is located in what we would call limbic or stress circuitry. This includes the amygdala and the hippocampus. Those are two of the primary regions that we need to know. And these are regions involved in emotion and memory, and also the modulation of our stress system, of our HPA axis, and our regulation of our hormones, actually, stress hormones, are very related to the 5-HT1A receptor and the hippocampus in particular. So this model of the 1A receptor, sorry, there's like multiple lizards coming towards me in this room right now. They are thought to relate to a variety of processes that enable us to passively cope with stressors in our life. And what the research supporting this is that when you activate the 1A receptor, people become more patient, less impulsive, less aggressive, and less anxious. So when you think about it, when somebody's really stressed out, they might be impatient, they might be a bit more impulsive, might be a bit more on edge, anxious, and so on. So activating this receptor actually decreases all that and really kind of calms people out. And this is the way that's predominantly, this is the receptor that's predominantly targeted by conventional antidepressants like SSRIs. And so in 5-MeO predominantly hitting this system, it means it's very strongly activating this system and therefore likely in boosting neuroplasticity specifically in regions related to regulation of stress and emotion. Whereas serotonin 2A receptors are thought to underlie a more active way to deal with stressors in our life. And there's research suggesting that it makes people more flexible in their behavior, increases the plasticity of their brain and behavior, makes them more sensitive to the environment that they're in, enhances their ability to learn and to adapt. And so it complements this 1A receptor by, instead of just kind of relaxing yourself and chilling yourself out to deal with stressors, it's motivating active, dynamic, flexible, adaptive responses to then, handle them in a more action-oriented way. And this is the one modulated by serotonergic psychedelics. And this is located more throughout the cortex, but actually is particularly rich in the default mode network. And so in virtue of that, serotonin activating this receptor, which again, psilocybin, DMT, and LSD do, you're getting more plasticity in the default mode network, which is involved in parts of our brain function or like cognitive function related to our sense of self, our kind of identity of who we are, our story, our narrative, and also just how we conceptualize and interpret our experience. So those regions are more altered by the serotonergic psychedelics, which target these receptors. Whereas again, 5-MeO predominantly hits the 5-HT1A, which is again related to things like stress modulation and memory. But I do want to remind you that 5-MeO does hit both again, but the 1A more, which is quite interesting. And so there's actually only been 2 studies to date that have specifically looked at 5-MeO and neuroplasticity. one of them did this interesting thing where they used stem cells to create these mini brains called cerebral organoids, and basically put them in a dish and then put 5-MeO on them to see what happens, essentially. And they analyzed gene expression and protein expression. And they found that in these mini brains that they created in the dish, over 900 proteins were changed in their expression after DMT. And then they analyzed, okay, so what are these proteins for? And actually the majority, or like many of them, were related to structural and functional plasticity, mostly related to things like increasing synaptic strength and increasing dendritic complexity. So making synapses between neurons stronger and also making them more complex, so more neurons have a chance to activate a given neuron, which presumably can potentially underlie greater flexibility in that neuron and the behaviors that it's supporting by allowing for new possible connections. And then another one, a bit of backstory on this. They administered straight into the brain of mice, into a region called the dentate gyrus. And so here's an important distinction between neurogenesis and neuroplasticity. So the dentate gyrus is one of the few, or maybe perhaps the only region in the brain, in the adult human brain, that creates new neurons like totally from scratch. That's neurogenesis. And neurogenesis is very different from neuroplasticity. As we've been describing it, it's already existing neurons, how they connect to each other. Neurogenesis is the creation de novo of a new neuron. And this seems to only happen in regions in the hippocampus, such as the dentate gyrus. So in the study, they're asking the question, if we put 5-MeO straight into the dentate gyrus of the hippocampus, does that boost its tendency to create new neurons? And also, what are those neurons like when that happens? And big surprise, when they did this, it actually boosted neurogenesis quite a bit. And so there's an increased number of neurons being created in this brain region. And what's interesting, and that was in adults, when they tried it in newborns, they found that those neurons in the dentate gyrus exposed to 5-MeO matured much faster and were much more complex than other neurons not exposed to it. So it's kind of accelerated these processes that lead to the ability of this neuron to receive and interface with many other neurons, which is essentially boosting its structural plasticity. And it also basically lowered the amount of input neurons in the hippocampus needed in order to fire, to make them more easily able to fire, which is functional plasticity. So in general, from these two studies, with 5-MeO in particular, it suggests that it does strongly boost neuroplasticity by changing the expression of proteins within neurons that are conducive to making neurons able to connect with, to form potential possible connections with a whole variety of other neurons, and also increases the ability for different neurons to fire each other, potentially in creating efficiency of neural firing, and also potential for new connections. And again, this doesn't last forever. It's unclear on how long this boost lasts. But again, the way that the brain works is if you fire given neurons together over and over again, it's going to make that synapse stronger and more longer lasting. So therefore, you get this boost in neuroplasticity, you may get more neurons in your hippocampus, more possible connections. But again, what you're doing with them is what matters. It's the integration and the consistency in actions to support the new behaviors you want to encode. And so just for comparison, or just like to broader, to talk about this, there's more research with the other psychedelics, LSD, DMT, particularly have been focused on a lot. Also, this other compound often used in rodent research called DOI, which is also activates the serotonin 2A receptor, and it's kind of a easier to use research compound. I believe it's used more commonly because there's less legal restrictions around it. And so it's more easy for researchers to obtain it. So there's a lot of rodent research and neuroplasticity research related to psychedelics done with this compound called DOI, even though humans, you don't really hear about people taking DOI. And if you look at this image on the left here, this is a placebo, so they didn't really receive anything. And you can see with DOI, with DMT, LSD, you can see how these neurons are becoming more complex. There's more branches, which again are either receiving from other neurons or activating other neurons. And this is an actual, in the middle here, an actual image of the neurons. You can see this is presumably the axon of it. And, or it could, no, it's more likely a dendrite. This is a dendrite. And here with DOI, you see some protrusions coming out and these little orange dots or spines, so areas where other neurons can connect to, you get more with DMT, and you get a lot more with LSD as well. And then, quantifying this number of spines, which again is like you could think of it as a landing pad on a given dendrite to receive an input from another neuron. You get significantly increased number of spines per 10 nanometers or micrometers. And in terms of the density of synapses, so the number of connections between neurons, in terms of a fold change, so like a multiplier, so like 1.3 times change, 1.8 times change, so it's higher with all three of these drugs as well. And so we know pretty firmly that these psychedelics do increase structural plasticity and facilitate the formation of more synapses, more potential connections between neurons. And again, this dovetails with the 5-MeO, because 5-MeO is structurally similar to these drugs and very likely, would do the same thing if it was included in the study. But again, probably in different parts of the brain because of the receptor difference. This is a really cool figure from a review paper, because obviously a pertinent question is how long do these things last? So let me walk everyone through this. So these are different processes related to neuroplasticity. People might have heard of BDNF, which is brain-derived neurotrophic factor. So it's here, brain-derived neurotrophic factor protein increases at 4 hours and seems to last up to 2 days. And so this is a protein or a chemical essentially in your brain, which signals, initiates signals that boost neuroplasticity and boost dendrites, etc. And so that comes online about 4 hours. And then at 6 hours, then you start to get dendritogenesis, synaptogenesis, spinogenesis. So genesis means creation of, dendrito, dendrite, synapse, spines. So the rate of these starts to increase, and then the density is found to be increased in one study up to 30 days later. And so it starts at about 6 hours. So for example, if you took some psilocybin, it's toward the end of the experience actually that your plasticity is primed. So like that last few hours of your experience, and then, predominantly the next 3 days is when your brain is hyperplastic with all these potential new connections. So that's when, it's really essential to be careful what you're doing, what you're putting your attention on, what habits you're engaging in, etc. It's really, again, from 6 hours from ingestion to 3 days. And it's interesting to think about 5-MeO here. It's not entirely clear. This is mainly based on research with psilocybin, which is, again, a very longer lasting. And so it's interesting to see, and we don't know yet, how the length of the neuroplastic changes from short-acting psychedelics like 5-MeO last maybe 10 to 20 minutes, and then DMT, which lasts 10 to 20 minutes. If those neuroplasticity boosts, which are potent in the short term, last as long as these other longer-lasting drugs. And we just don't know that because there's just not enough research yet. And just takeaways, again, 5-MeO works quite differently compared to other psychedelics. It is a potent booster of neuroplasticity, but likely in different regions because the serotonin 1A receptor is located in different regions than the serotonin 2A receptor that other psychedelics hit. And it seems to affect the hippocampus more than the cortex because hippocampus is quite rich in 1A receptors. And again, hippocampus is this very complex region involved in memories of past experiences, our ability to create associations between things. And it's also very closely linked to our stress system and our endocrine system, which modulates the release of hormones such as cortisol and other stress-related hormones. And again, as I just said, open question, how long do these last? Most likely, most potent from 4 or 6 to 72 hours. And again, critical for integration. But all that said, again, there's only 2 studies specifically looking at neuroplasticity with 5-MeO to date. And presuming there's tons being done in the next year, 1 to 2 years, there's going to be a lot more research on it. But for now, we can kind of infer that the research with the other psychedelics broadly is similar to 5-MeO, but the nuances there need to be disentangled. And yeah, that's all I got for slides. So thank you for your attention.
Awesome. Thank you.
Thank you so much.
That was very beautiful. Yeah, very comprehensive. Thank you for laying that all out, Manesh.
My pleasure.
First up, we've got one question that was just from a few minutes ago, and regarding the mini brains And Mr. Benjamin was asking, do these mini brains express both the 1A and 2A receptor types equally?
Yeah, that's a really good question. I believe they probably not, actually, because usually in most of the regions of the brain, they do overlap, 1A receptors and 2A receptors. And these mini brain organoids are just very small replicas of the brain, and they're just they're usually engineered with a particular subset of receptors. So I actually don't know the exact distribution of each of them in there. That's an interesting question, but it would be relevant. Yeah, I have to think because what's interesting, and I didn't touch on this too much, is that the 1A receptor, when it's activated, it makes neurons less likely to fire. Whereas the 2A receptor, when it's activated, it makes neurons more likely to fire. So there's an inherent competition or a balance, that like a dance they do with each other to balance out to see who wins. And so in certain contexts, one will go above the other. And so in this case, regardless, even if the distributions were equal, if 5-MeO is there, it's going to activate the 1A more than the 2A. So that's going to take dominance. So it's going to all have an overall inhibitory effect. But it's interesting. I'm not sure actually which one in the study was expressed more.
Does it in full doses, does it also become a full agonist of 2A?
Okay. Yeah, it's unclear like to what extent it will. It's just that it has a natural affinity to go to 1A receptor. And for example, one finding suggesting that is that maybe you've noticed this in people who've come through Tendava, is that at lower doses it induces hypothermia, so people get colder, but at higher doses people get warmer. And what's interesting is that the 1A receptor specifically has been related to being colder and 2A to being warmer. So that process itself suggests that there's a change in mechanism happening at some stage where people start to feel warmer, because that's the 2A is becoming aligned to a significant degree. But to what extent, and whether that's an extent comparable to psilocybin and these other drugs, there are ways to do this with brain imaging, but it just hasn't been done yet.
Gotcha. Yeah, hopefully we'll be able to get some more juicy information. As you know, we have the upcoming studies with UCL here that we're hoping to launch in either August or September, but we'll be getting the peak 4 to 6 minutes of the 5-MeO experience with a 64-point EEG. And then there's a few other potentials, but what we were just talking about dovetails perfectly into another question here, and I'm pretty curious about this as well. Mr. Benjamin was asking, do you think the non-dual state that comes with the full release comes from the 2A receptor at a high dose, even though 1A is more involved in the default mode network?
Yeah. Well, first I want to say the 2A is actually more involved in the default mode network. And in some cases, the hippocampus is thought of as part of the default mode network as well, which is interesting. So, 2A receptor densities are highest in the default mode network everywhere except for the hippocampus, and the hippocampus is full of 1A. So that's like an interesting thing. But yeah, it's really interesting. I think 1A and 2A both contribute to these kinds of states. I think, often with 5-MeO, there's a strong body load. There's a sense of kind of being immobilized, and you just lose touch of your body, and you're just so almost like neutralized in terms of being relaxed and grounded downwards, if that makes any sense. And that's a very 1A effect, because 1A calms, it stills, it relaxes. And so that initial kind of calming, kind of like almost sedating relaxation effect at the beginning, that's very 1A. And that can also lead to kind of internalization of the senses, right? And that's often associated with these kind of ego dissolution non-dual states, because then you're not in tune, in terms of an internal sense of it, having this internal void within yourself. You having this like almost pratyahara type thing where you're losing sense of your external surroundings, and that's very 1A. I think 2A is more so losing all sense of who you are conceptually, like the concept of Manesh going out the window. I think that's more of a 2A thing. So that's why people can have the strong body load and have this intense experience, but they kind of know who they are still. And at higher doses in a full breakthrough, you lose that. I think that's the 2A coming online.
Gotcha. Interesting. That's really cool to know. Here at the center at Tandava, we work with both low, medium, and high dose protocols depending on each participant. And, we utilize low and medium doses quite often. Particularly, we find the most effective to do actual work, if we're working into specific areas, into specific patternings. Yeah, so that's very interesting.
Mm.
Some more questions, and just to answer a few real quick. Yes, Adam, this will be recorded and available for replay by before next weekend and probably the next few days. And then Charity, no, the recording will not be sent out in the email, but it will be made available on the 5 YouTube page. And then Brother Byron asks, are there other intracellular receptors modulating the molecular effects of 5-MeO?
Yeah, definitely. So I didn't get into the details of how neuroplasticity is elicited exactly, but when there are things that happen inside of a neuron to give rise to these changes in dendrites and things. And usually it's through BDNF being produced, and this influencing other downstream processes, including this thing called mTOR, which is another, I guess it's a protein that changes the ability for certain receptors to be increased at the synapse, and also for dendrites to grow and change in different ways. And so, and the other gene expression changes, some of which we don't fully know or have uncovered in detail yet, all happening inside the receptor. Because in order for the, inside the neuron, in order for the neuron to create dendrites, it has to translate proteins for those, right? It's creating new materials that usually it wasn't creating. So you're turning on different parts of the genetic code inside the cell to create these new things. And so there are a variety of intracellular genetic, like epigenetic changes happening, which are mediated by things like BDNF and other compounds.
Gotcha. Awesome.
Amazing.
Thank you. Let me see. Whoop. Benjamin says, awesome. Thanks, Manesh. Big question from me. Have you seen the psychedelics promote neuroplasticity through the activation of intracellular 5-HT2A receptors? This was a quote. Can you comment on the role of intracellular 1A or 2A receptors in plastogenesis of 5? Does this explain how endogenous 5 may lead to plastogenesis?
This is really interesting. Yeah. So he's talking about this recent paper that came out, which basically said it was mainly about the 2A receptor. Well, it was about the 2A receptor in particular. And they found that because obviously the serotonin 2A receptor, 5-HT2A receptor, gets activated by serotonin because that's naturally the compound that activates it, right? But they found that what was different about psychedelics is that they go inside of the neuron and hit 2A receptors inside of the neuron, and that's what boosts neuroplasticity. And when they altered the serotonin to give it the ability to go inside of neurons, it actually had psychedelic-like effects on boosting neuroplasticity. And the interesting thing about DMT and 5-MeO-DMT is that they've been found in human urine and blood samples. So they're endogenously created somehow. And so it is possible that these are created and are able to induce these endogenously created neuroplastic effects because they're able to go into neurons like serotonin can't. So I think very likely 5-MeO through its effect. We don't know if on the 1A receptor if it does similar things, but on its effects on the 2A receptor, I'm sure similar to other psychedelics, it's doing that in a way different than serotonin because it's getting inside of the neuron. And again, it's particularly fascinating because, what is 5-MeO and DMT doing in our brain? And when is it actually, in sizable enough amounts to induce an effect? These are all things we don't know about, but it is interesting. It fundamentally works different than serotonin. And why would these receptors exist in the cell if there was nothing that's coming in there to activate it? And it's not serotonin. So, it's a bit of an interesting mystery there. And I'm sure many scientists are on it right now.
I love it. All right. So Brad says, so 5-MeO acts like an inhibitory neurotransmitter on the limbic system, calming an overactive limbic system?
I think so, yeah. I think it really just basically shuts it all down temporarily, and that's part of the experience. So it really dampens it and potentially allows for this resetting effect for those kind of overactive circuits to then kind of turn off for a bit and then come back at a recalibrated level. I think that could be part of it for sure.
Yeah. And then Byron was just commenting or asking and commenting something you were just saying, and he says, yes, they're too complex of mTOR, sir, but I was wondering about the sigma receptor and even intracellular receptors of serotonin.
Right. Yeah. So we just talked about the intracellular from Benjamin's question, and sigma receptor, there's no evidence that 5-MeO hits the sigma receptor actually, and then DMT does. which is another way, and then DMT boosts neuroplasticity and also might be particularly relevant for addiction, actually, because sigma receptor is another receptor in the brain that's located in addiction-related circuits, actually. And I believe there's a study, for example, where they blocked the sigma receptor and it made it harder for mice to become addicted to cocaine. Very interesting.
Wow.
But again, there's no evidence that 5-MeO hits it like the other DMT does.
Gotcha. And Benjamin says, so the intracellular 2A receptor drives the DNA transcription and then the protein translates that drive the plasticity. The intracellular receptors could be significantly different in structure so that the intracellular could be more potently influenced by 5.
So first question, yes, that's basically what we understand. This happens. Yeah, that activating these cellular 2A receptors leads to different epigenetic changes that then drive plasticity-related changes. And the intracellular receptors could be slightly different in structure. There's a couple of ways to understand what you're trying to say, but I think one of the ways you're asking if we can modulate intracellular receptors to make them more influenceable by 5. In principle, that's probably, but but again, it's not even clear if 1A receptors, which 5 predominantly hits, are intracellular in the same way that 2A are. But if you're talking about 2A activation by it, oh, I understand this question now. You're asking if that's like a little hack to get more 2A activation with less 5-MeO by making the intracellular receptors more sensitive to it. That's an interesting one. It's possible. It's in principle possible. I'm not sure how it would happen, but at the end of the day, no matter what, 5-MeO will gravitate towards 1A more in general. But it's possible you can get more, more for your money's worth of 5-MeO from the 2A with some changes, but that would have to be in mice by changing the genetics of it, which we can't really do in humans.
Benjamin says, thank you. He says, no, I just mean that the pH intracellularly could change the shape of the 1A or 2A intracellularly, even though they come from the same gene as the receptor in the plasma membrane. He's doing some R&D for a pharma company, aren't you?
Yeah, these are some very specific questions. I like it. Could change the shape of the 1A to the pH intracellular. The easy thing I could say here is I don't know. I mean, it's interesting to think about the different ways that the 2A receptor can be modulated to increase its sensitivity. And there is actually evidence that serotonin 2A receptors can be in a high affinity or low affinity state, that they could change into the degree to which they attract serotonin based on certain activity patterns. And so there are switches you could flip in relation to the serotonin 2A receptor, which make other neurotransmitters more likely to go to it. And so these are things that can be explored in rat models by, changing genetics and seeing the underlying kind of factors that contributed to it. But definitely we don't really know at this point. More research is needed.
Awesome. And let's see, do we have any more in here? I've got a question. Manesh, I'd love to know, what are some of the possibilities that can come from gaining a deeper understanding into all of this? For example, where do you see biotech going over the next 5 or 10 years? What could all of this allow us to understand and do? Kind of some cool things that you see popping up and happening?
For sure. I think this interest in 5-MeO and the other psychedelics has really stimulated an interest in what we can call transdiagnostic mechanisms, right? Because in the past, you design a drug for a particular thing. You design a depression drug. You create an ADHD drug. You create a drug that's better for people who have anxiety. And things like psychedelics, they work for so many different disorders. And for a variety of reasons. One, because many have an underlying basis in different forms of trauma and developmental insults or like needs not being met, et cetera. But also this neuroplasticity thing is a huge one. And so there's a huge emergence of neuroplasticity focused therapeutics. And ketamine is one of them, and other things based in glutamate. But these psychedelics are also another major one, and people are using that. People like the Olson lab at UC San Diego, I want to say, somewhere in California, they're all about using psychedelics and how they work as a jump off point to creating more psychoplastogens, more compounds that tap into plasticity. And so I think plasticity is going to be a major theme in kind of pharma, basically, and also in kind of behavioral interventions more broadly. It's like, how can we enhance neuroplasticity and kind of encode the changes in a lasting way? And I think that can help target a whole variety of disorders. So again, we're moving away from, I think, focus on specific ones to find the general patterns, and neuroplasticity seems to be a big one. And it's going to be really interesting to see how that plays out, because they're also trying to take out the trip, right? They're trying to, like, so they ask, oh, psychedelics are so good at boosting neuroplasticity, but they also give you this trip, which to be fair is not good for everybody, and not everyone is ready for it. So it's like, how can you get drugs that do these things without having the investment of 8 hours of your life, or even in a very intense 5 experience, for example?
Yeah.
Interesting. I love it. You have any questions?
No, I don't. That's been an incredibly informative webinar. Thank you so much for coming on to share your wisdom with us.
Yeah, my pleasure. Totally happy to do it.
Yeah. Awesome. Well, looking forward to catching up with you again, Manesh, and whether it's here in Mexico or somewhere else, or at a conference somewhere. And
yeah, 100%.
Yeah. Thanks again for coming on. And everyone, thank you for joining us. Everyone, thank you for your great questions, and thank you for engaging. And yeah, really appreciate it. I hope everyone swings by the 5 site, 5-MeO.education. And all of our talks will be up on there. We've got talks with Hamilton Morris, with Dr. Bianca Sebin. Our talk with Manesh will be up in the next couple of days. We had one with Ian Benwee speaking about silomethoxin last month as well, or 2 months ago. And yeah, stay tuned to the 5 platform. We'll have some fun stuff going on from there, some fun announcements coming up soon, some very fun research happening soon. And as always, we feel that, but we really believe in collaboration over competition. And so anyone who's ever looking to do something really cool around 5-MeO-DMT, this 5 is around the community. It's not about our company. It's not about Victoria and myself or any of the contributors. It is about the medicine, and it is about the community. And we are all about bringing everyone together. So we're hoping to see you all on the path somewhere, whether on person or online. Lots of love to everybody. We'll have this posted, as I said, in the next few days. People were asking, what is the website? It is 5 as in FIVE-MEO.education.
Yeah, and if you're looking for the recording of the webinar, go under the tab that's about us, and you'll find the event recordings there, and it'll be up by next Sunday for sure.
Yeah, awesome. And yep, we hope to see you all soon. Thanks again for signing on, and thanks again, Manesh.
Thank you.
Thank you, guys. Have a great
Ciao,
night, everyone.
Have a great one.
Bye.
Take care.